Hi, so welcome to a new topic, and this is beta-lactam antibiotics. Very important topic and very important group of antibiotics. So why I call them as beta-lactam antibiotics? Because all of them, they have a beta-lactam ring. So that's why they call us beta-lactam antibiotics. Now, various types of beta-lactam antibiotics are there. The first one being the penicillin. The second being the cephalosporins. The third being the carbapenems. And the fourth one being the monobactam. So we are going to go through all of them one by one. So first, let's get started with penicillin. So penicillin, I will be actually writing most of the times at this symbol, which is PN, which is the short form of penicillin. So let's see what is the mechanism of action of penicillin antibiotics. Now these drugs, actually what they do is that they are going to act on they're mainly active against the gram-positive bacteria. And let's see why not against gram-negative bacteria. So first we'll see how they are going to act, and then we'll see why they're not active against gram-negative bacteria. So you see, this one is on the left hand side of the screen, it is the gram-positive structure. So they have a cell wall. Yes, they have a cell wall and they have a cell membrane. Now cell wall, they are comprised of various peptidoglycan rings. So these are the peptidoglycan chains, and they are linked by these green lines, which are the cross links. So, if you can imagine, this is giving them strength. So if you imagine, a cobweb by the spider, when he makes like web like this, and beech may again he, puts cross links in between. And why he does this? Because it strengthens that cobweb. Agar wo nahi hoga, it's gonna break easily, this cobweb. So similarly, the cell wall strength is because of this interlocking between the various chains. Agar wo nahi hoenge chains, then the cell wall becomes weak enough and the bacteria will die. So there is cross-linking is very important for the integrity or the strength of the cell wall. Then we have the cell membrane, and in the cell membrane we have these yellow structures that are called as penicillin binding proteins. So as the name suggests, penicillin binding proteins, that means the penicillin are going to actually go and bind to it. So what does the penicillin do? Let's say this is the penicillin here. It is going to go through the cell wall and it is going to bind to these penicillin binding proteins here and here. Okay? So it is going to bind. The site is the penicillin binding protein. So when it binds to these penicillin binding proteins, what it does is it basically has the aim of targeting the cross links. It is going to break these cross linkages. And how does it do that? Let's see. So ये जो white cross links हैं, they are made up of the peptidoglycan chains, जो white structure था, it is made up of NAM and NAG. ठीक है? So and NAM and NAG is present, and they have so many amino acids, they're linked to them. So with alanine, glucine, lysine, alanine again. Okay? So those white structures which I showed you here, which were like this and this, so they are actually made up of these components. And now we are going to talk about how the cross links are formed. So let's see what is the normal process of forming of the cross links. So when these two white structures, which I told you, they are comprised of, this is the structure of that. Normally what happens when they're actively multiplying and, the cell wall is also getting synthesized, because the terminal D-alanine, these are two D-alanines here, 1 and 2. So the last D-alanine, it gets broken away or it gets cleaved off. It will just removed from this whole peptidoglycan, the chain. And the enzyme responsible is transpeptidase. So transpeptidase, what it does is, it cleaves, काट देता है. It cuts away the D-alanine from this particular site. So it cuts away this D-alanine. Now the bond is broken. Okay, so this is cut away, right? So this is broken bond, it is cut away. And when it gets cut away, there is release of energy, right? And that actually is utilized in formation of a bond, and that form is bond. See this L here, you see this L-lysine has 5 glycine units attached to it. So what they do, this 5 glycines here, once this transpeptidase enzyme has cut off, this is this here step number 1 is it has cleaved off this D-alanine from here. So now this 5 जो है हमारे glycines, which are 5 in number, they form a bond with this D-alanine. So they are cross-linking these two. So this green one structure is actually this here. So on and so forth, आगे आगे भी जाके same, ठीक है? So यहाँ पे there is another NAM chain attaching all these amino acids, and then this is going to form a with the other one. So this is how the cross link is formed. Okay, so this is how the strengthening of the cell wall of the bacteria is going to take place. Now what does our penicillin do? Now penicillin, the role of penicillin here is that it is going to inhibit the enzyme it binds to penicillin binding proteins, and it inhibits the enzyme transpeptidase. So when it inhibits the enzyme transpeptidase, the cleaving, the first step, which is important, is not going to happen. D-alanine is not going to get separated, and when D-alanine does not get separated, there is no release of energy. This bond is also not being formed. When the bond is not being formed, so there is no cross links between these two, right? So if we go back here, ये जो bonds है, they get broken. They are not formed. All these bonds are not formed. So when these bonds are not formed, this whole thing, it becomes very weak. The cell wall becomes very weak because there's no, this because it's called crumble, right? Like this, there's no nothing which is holding it. So it becomes very weak. The strength is lost, and when it becomes so weak, what happens? The water from surrounding, a fluid from the surrounding, it is going to enter. So the fluid, let's say यहाँ से, the fluid is going to enter, and this is going to make the cell really big. It is going to the cell is all going to be filled with this, and it's going to swell up. और जब ये swell हो जाता है, ultimately this will burst, and obviously it is dead. So it is bactericidal. It will kill the bacteria because it damages the cell wall. Cell wall is very important for its survival. If there's no cell wall, it bursts, then the bacteria is going to get killed. So remember, the first what it does, it is going to enter into and bind to the penicillin binding protein. Then it's going to target basically the cross links, and cross links के लिए important है the enzyme transpeptidase, because it cuts the terminal D-alanine, and then there is release of energy which will lead to formation of this bond, okay, which is this bond. And when there is no transpeptidase enzyme, no breaking of the D-alanine, this bond is not formed. And when this bond is not formed, there will be weak cell wall, and weak cell wall means they're going to burst because a lot of fluids etc. is going to enter inside. It's going to lead to cell swelling and burst. And also there are certain, some penicillins also decrease some important lysines, and they kind of destroy this. So this is the main mechanism of penicillin, and this is why they are effective against gram positive. Now why not against gram negative? Let's see to the right side of the picture. You see right side pe gram negative ka structure. They have a cell wall like penicillin, check. They have a cell membrane like penicillin, gram positive, check. But what they additionally have, they also have a outer membrane. That's an additional feature which this was not present with the gram positive. So ये जो outer membrane है, it consists of gaps in between, which is called the porins, and also they have some enzymes like beta-lactamases. So the first problem faced with the drug is that now it has to travel a big distance from here to reach the penicillin binding proteins. यहाँ पे उसको सिर्फ यहाँ से यहाँ तक जाना था. It has to travel so much, it becomes difficult for it to reach inside the penicillin binding proteins. Then these porin channels don't allow them to go inside. The outer membrane is actually a covering, protective covering for them. They don't allow these drugs to enter inside. And the third thing which happens are these enzymes, they destroy the drug. So therefore, gram-negative, they're naturally resistant to penicillins. This penicillin, especially the benzyl penicillin, the natural penicillins. So 3 reasons. 1, because of the outer membrane becomes difficult for the penicillin to enter inside. Then the porins, they are not allowing it to move inside. And then beta-lactamase is also present, which are the third problem with them, because they can damage or destroy the drug penicillin. So therefore, they are not effective against gram-negative, only against the gram-positive, right?